Okay, so welcome to another episode of the Biohacker Blondie Podcast. I'm really excited for this one because we're here with Dr. Oliver Zolman. And we actually met in Los Angeles. And then we had the most amazing conversation in London over dinner for 4 hours. And now we're here in Miami. If you do not know Dr. Oliver Zolman, he is a longevity researcher focused on reversing aging across all 82 organs in the body. He is known for co-leading Project Blueprint, one of the most extreme and data-driven anti-aging experiments in the world, and for developing the level 1, 2, 3 longevity framework aimed at extending human health. And it keeps going on. Founder of Longevity School and pioneering research in gene therapy, triple helix gene therapy, which we'll get into, organ rejuvenation, lifespan extension, and including your ambitious project, including Project Mousespan. So thank you so much for coming on the podcast. There's definitely a lot we can go over about reversing aging and all the advanced procedures that are going on and how you started in this field of longevity and reversing aging.
Attempting to reverse aging, yeah. easier said than done. Well, during med school, before med school, I was always interested in stem cell biology, kind of nanotechnology.
In med school?
Before med school, yeah.
Before med
school, like in high school in UK.
Wow.
I was like super keen on all those things. But then I get to med school and none of this exists.
Wait.
And I'm like, oh shit. It's not as advanced as I thought it was in all 7 years of med school for me. So yeah, that was kind of my motivator. Like when I saw that gap between all the cool stuff going on in the research, preclinically in animal models or some, early stage clinical stuff around the world, that was that kind of like woke me up to how different clinical practices in the NHS in particular, in my case, versus like what the research is showing is coming.
Wow. So you were way ahead of the medical system.
Well, I mean, in terms of ambition, I guess, yeah. In terms of what I thought like you could do in my naive state, like when you're like, 16, 17, learning about biology for the first time.
So what intrigued you at that age to start looking into aging, longevity, reversing the aging? Because, I feel like the medical system was so behind. I mean, the brain gut axis, they're just saying, just recently, saying there's that connection, in medical school and everything. So, and we've always, especially in the US, it's like, we wait for disease and then we give a pill to cover up everything. So were you reading books? Did you listen to a podcast? Are you fond of David Sinclair? Like how did you get into that?
Yeah, I was doing an internship in the Weizmann Institute of Science, actually. And then I met someone there who was doing a conference in Cambridge, my hometown in UK. And she was speaking, I think, and then she invited me. And then I went to that conference, and that was Aubrey de Grey's conference. So then I read Aubrey's book, Ending Aging, which is so technical. It took me like, I don't know, it's like I spent a couple 100 hours reading that book.
Over and over?
Not over and over, just like very slowly. That's normally how I read like textbooks. I read them very slowly so I understand like every concept in each sentence, each paragraph, across the whole like textbook. It's kind of a textbook really, Ending Aging, Aubrey's book, rather than a kind of...
I need to read this.
Is
it up to date still, you think?
It's still like it's still up to date. It's still up to date. Like most of the concepts are pretty up to date.
Okay.
But that was like then I had this understanding. Then I had Aubrey's perspective, and all these different scientists and medical doctors at the Cambridge conference at Queen's College, and that kind of combined with my like interest already in stem cell biology, stem cell therapy, nanotechnology, etc. That then kind of gave me this perspective that there's this huge area of kind of biology that we're not able to tackle yet with the current medicine. Of course, we've had like age-related disease medicine for forever, with like most medical care is age-related disease medicine already, right? But it's always not that effective, unfortunately.
Yeah, it's not very proactive, preventative. It's more reactive. So
Yeah.
when you went into medical school, were you kind of like, I just want to get through this?
No.
Okay.
No, it was an amazing experience. You learn so much, and it's like so challenging, like not just kind of intellectually, but like physically as well, and like socially, with all the kind of like extreme interactions you have to have with patients, which you don't really, I don't know, unless if you're like a soldier, maybe that's kind of similar kind of you see similar kind of things. But there's not many like professions where you're exposed to so many like extreme and kind of emotionally challenging situations.
Yeah, I can imagine. So when did you get into kind of thinking, how do I reverse each organ?
Yeah, so oh, it took me a while. It's so obvious, right?
Aging and reversing and maybe living forever, it's very complicated.
Yeah.
And, I've been a fan of David Sinclair. I've listened to so many podcasts on this, and we, specifically like looking at individual organs, but knowing that your body is all connected. So it's kind of, there's so many interesting facts about longevity, like different animals that live longer. And then now you're looking into mice, but then it's like, okay, well, what about their body mass? Like all these things play a role into longevity and reversing aging. So you started to think, okay, let's see the age of all the organs.
Yeah, I think maybe like after 5 or 6 years of thinking about it, then I came to that conclusion. I was like, oh, to like reverse aging, you have to measure aging.
That's a good start.
Like no one seems to realize that. Especially at the time, it's a little more common now. But like when you think of like Alzheimer's, whatever, no one thinks about measuring brain age, or you're just measuring like p-tau 217 or PET amyloid or whatever, like clinical scores.
Yeah.
So yeah, it's like a very obvious conclusion that if we have to measure age to understand if we reversed it, and we have to measure it accurately. We can't just measure it inaccurately because then it wouldn't work.
I know. Is that
So we have to measure age
or is it
accurately.
Were you the one setting the standards for the aging of like how do you measure your brain age? Or I mean, were you at the forefront of that, of saying this is the typical for your organs and this is the typical for your brain age? And because I feel like this is all, you're right, this is kind of a new thing of saying our biological age. It's not, a lot of people know this now, like we can kind of round up all of our biomarkers and say, now our biological age is less or greater than, our chronological age. But how did you even come up with like where the biomarker stands for each organ?
Yeah, so there's two things related to this. So one is like, how do you measure age accurately in an organ? And then two, how do you in terms of like just the raw pathology of what's going on, like the molecules or whatever. And then two is like, how do you do that in a biostatistically accurate way as well? So they're two kind of interrelated problems. And yeah, and then at an individual level as well, because it's different when you do this in like a big study. You can get away with like, a lot of stuff statistically. But when you try and do this in an individual and really prove that you've sustainably made a change in their biological age of a particular something, that's more difficult than when you have 1,000 people or 100 people in a study and you can take an average and get away with a lot of stuff. So yeah, when so I'm very like a systems thinker.
Mm-hmm.
So like, okay, I was like, you have to measure age, but then we don't, all the organs are important.
Yeah.
So we have to measure age in each organ. And how many organ types are there? You know, you count them all up. I added one in recently because I forgot the fascia. Everyone forgets the fascia. Except the Chinese. I think they're quite big on fascia historically. So there's 82 types of organs across both genders. And to prove you reversed aging in the body, you'd have to prove you reversed aging in each of your organs. Very simple, right?
Somewhat.
Somewhat, right. So, and then, yeah, then you have, it's like, how do you, so then it's another way of systems thinking. Okay, so what type of ways of measurement are there possible within medicine or physics and chemistry and biology? So there's kind of 3 or 4 categories. There's biosamples, which is blood, urine, saliva, whatever, biopsy as well. There's medical devices, and then there's imaging-based markers, and then there's kind of questionnaires, which can be classified as medical devices, but you know, questionnaires or kind of subjective kind of stuff. So that's kind of like all the possible ways of measuring anything in biology. So that's the framework you have to work in. And then the more of those you add in, typically, to a given organ, the more accurate you can prove, more accurately you can theoretically prove you're reducing the biological age of that organ. So having like all types of those markers measured for a particular organ is probably going to outperform the ability to prove you reversed its biological age than just using one of those types of markers.
Yeah.
For like an extreme example between 2 comparisons. So say with the spleen, you just had an ultrasound of splenic artery resistive index. That's like one marker. But say you then, that's your one marker in your model of the biological age of the spleen organ. But then what if you add in a load of fancy MRI, which can look at the contents of the spleen spectroscopically, water-wise, scar tissue-wise, calcification-wise, just volumetrically as well. And then you have like a blood test, which can do a lot of fancy things related to, okay, these are spleen-based markers, and these are validated for how they change with age. And these are all the different aspects of the spleen which we're covering within this relevant biosample, which is the blood. So you don't want to do a spleen biopsy. That's not very fun.
Yeah.
So you want to use easy like real-world stuff, in humans at least. So what if you're combining that model now to have, okay, you got like a fancy multi-parametric MRI, often using deep neural networks or various AI machine learning models to bring out features that radiologists or normal software can't do. And then you're combining that with your blood-based markers and ultrasound. And then you have a nice model of biological age of the spleen.
Of just the spleen.
Same concept for every single organ type in the body.
Okay, so has anyone ever done this? Did you first do this on yourself? Or I know you worked with Bryan Johnson as his doctor. Was he your first specimen for doing all these reversing 82 organs?
Right. So we're kind of at a point now where I think it's a lot easier to do this.
Okay.
Because of
Seems very complicated to do all 82.
Exactly. It's really like, it's a multi-person, full-time research project job just to do it on one person. Yeah. So you need so much MRI. You need so much ultrasound. I have like literally 1,000 pages, maybe 500 pages of manuals on how to do it.
Oh my gosh.
Because you have to like ultrasound, you need like 10 specialists.
Yeah.
Just within MRI and ultrasound to like really like even get the data and then process it into something that you can then analyze as a biological age. So it's not easy, right? But now we have LLMs. So
that's been helpful.
I've kind of been waiting for the LLMs to get just good enough to kind of restart this project, because yeah, it was very tricky to. It was too hard, basically. It was too hard before.
Yeah.
But now the LLMs, I can get a lot of, the next generation coming through, the next few months, I think is going to be the sweet spot. That's what I'm waiting for. Kind of the next generation of Claude, the next generation of probably Gemini as well. They're my favorite too.
Okay.
For this kind of stuff. So.
Do you have an Apple Mini?
No.
Okay, you're not there.
I got Perplexity Max
Programming,
with
just doing all the data for you, all your agents.
Well, yeah. I mean, you could use OpenClaw for it. I mean, yeah, maybe use OpenClaw. But I'd probably just you could use OpenClaw, but I might use Claude Code or Perplexity Max, Perplexity Computer. Is what I've been using currently for stuff. But maybe this kind of project you could do a dedicated Mac Mini. Which might have some advantages, but yeah, because these agents are getting so good now, they can run a lot of this machine learning statistics on that I didn't have time to do because there's so many specialties that you need. I need like a team to do this.
Yeah.
But no one wants to fund this project.
Yeah.
No one wants to fund this for whatever
Not
reason.
the pharmaceutical companies.
Well, I don't know. Like no one. No.
Do they?
I don't know.
They have the money, right?
I know hundreds of rich people. No one wants to fund biological age.
I wonder, okay, so what would be their, what do you propose to them that you're going to extend longevity up to 200? What is the main goal?
First step is just being able to measure age accurately in each organ. Okay. That's like
do you think this can be something that will be easy and maybe I can do in a day? Because that's the thing, if it takes so much time and money for one person, eventually do you think this will get to a point where most people can do this? Yeah.
Because of these, so the ideal situation, right, would be like you do a 1 hour MRI, you do like a 1 hour ultrasound, you do like a 1 hour medical history, and then you do a blood test and maybe a urine test or saliva test if necessary as well. And that would take like 3 hours, right? If you wanted everything. Yeah. And then because the AI is so good, you don't need to do like 100 hours of MRI. Okay. You only need 1 hour. You don't need to do like very complex ultrasounds that take many, many hours. Same situation, you only need like 1 hour or so. And then the blood tests, there'll be a lot of like, hopefully omic-based markers where you don't need 100 vials of blood, which can then, that just even taking 100 vials of blood is going to mess up your accuracy in a lot of things potentially as well. So you really got a limit on how much blood you can take, how much time you have for MRI, how much time you have for ultrasound to make this scalable.
Okay, so that's the main goal.
That's the goal, yeah. Okay. But I don't mind spending 100 hours on doing MRI for one person right now because like it's still kind of a proof of concept stage.
So did you start doing this on yourself?
Yeah, well, I started on myself, my parents.
Your parents?
Yeah.
Guinea pigs.
Literally, like my mum in an MRI scanner for the first time.
You're like, let me take more blood from you every day.
Literally, yeah.
Then like pump you up with some iron, keep the blood going.
literally, yeah.
So, okay, so were you testing all your organs, MRIs, ultrasound on yourself initially?
So some of them are harder. So some organs they don't really age much.
Okay.
Until like a certain age. Like they're harder to detect in various ways. Yeah. So like it wouldn't really make sense in someone like me. Okay. So this is why. So now that going back to the agents and them being able to do a lot of this AI model analysis in machine learning to bring out these regression equations that I can run to do this, these like very accurate biological age clocks at the organ level. So now that the agents can do that, I can take on another client who has a huge budget that wants to do this. Yeah. But who is that ideal client? It's someone that's older than Bryan, right? Because a lot of it's
Like you want someone 60 or something?
65 plus, probably.
Wow,
Yeah.
okay.
And ideally one of each gender, because it's kind of different. You're missing a lot of organs otherwise.
Yeah, I was going to ask about that, those reproductive organs.
Right, exactly. It's only half the problem if you're a man. So yeah, 65 plus, both genders, kind of average health, not super, super healthy and not like super, super unhealthy. I think that's kind of what would be a good next guinea pig.
Okay.
Because the kind of resolution you can pick up on biological age change, it's a lot easier to prove that when you're already 65 plus.
Yeah.
Whereas going from 45 to 40 or 35 to 30 or 25 to 20, that's very hard to show in a lot of organs.
Yeah.
Because
so when did you
you're so young already.
Get involved with, did Bryan Johnson reach out to you and say, I love this idea, concept, let's do it?
Yeah, a friend connected us, and then, yeah.
Did you do all 82 organs with him, or almost?
So, spleen we didn't.
Yeah.
Spleen, I might have got the resistive index ultrasound, but it's not a great marker for spleen. Yeah, like a lot of organs couldn't. They're not ready.
Okay.
They're not ready. I'd say about half.
Okay.
Whereas half we've got some like good stuff on.
Were you excited to be doing all this, like all every day, testing all these organs? The money's there, the doctors are there, everyone's just like, I mean, was that kind of like your dream, just being like studying this every day and having Bryan Johnson as kind of like the guinea pig?
That was my plan. Yeah.
So tell us the organs that you think matter most. Like if you had to, I'm assuming the brain, the heart.
Brain's pretty important.
Yeah. Reproductive organs. Have you done any testing with that as well?
Yeah.
I want to see the 65-year-old reverse the reproductive organs back to 20.
Yeah. So like sperm is quite easy to measure. There's like quite a bit of research on that.
What is?
Sperm.
Oh, okay.
Yeah, sperm itself. Whereas like, ovaries, they're a little harder to access. So they're a little harder to measure, and they also age very fast.
Really?
The ovarian organ, compared to
like
ovaries
after,
and thymus are some of the fastest aging organs.
Oh, interesting.
You kind of burn out by age, 45, 50 for ovaries. Same with thymus.
Wow.
Becomes,
I didn't even think of the thymus.
Turned into fat mostly, and you
what if
lose
I take thymus and alpha 1?
We'll have to measure your organ age to prove whether it is getting younger.
There's so much to test.
Rather than random acts of biohacking.
I'm telling you, I'll be your guinea pig. Just pump me up with thymus and alpha 1 and test my thymus.
Well, tesamorelin, promising.
Okay, I love tesamorelin. That's one of my favorites.
Oh, really? You tried it?
Yeah.
Yeah?
Yeah, that's
What'd you feel?
It's just like visceral fat, but like I feel great on it. Like it's one of my favorite peptides actually.
Nice.
Yeah. So, but thymus and alpha 1 was my intro with Dr. Matt Cook.
Right.
But that was like, I had mold and parasites and candida and you name it.
Yeah.
And was also really good for COVID. I took it during when I got COVID as well.
Oh, cool.
Yeah.
Yeah. Interesting and understudied. But
so what's your favorite? So
in terms of the most important organs. Yeah. So
because cardiovascular health is extremely important as a leading cause of death.
Yeah. But then the cardiovascular health can be from kidneys. It can be from bone marrow. It can be from like testes or ovaries as well. So it's all like that's what kills you. But like the part of the reason it starts aging so bad is because your other organs are aging as well. So it's all interconnected.
That's the hard part about longevity and reversing aging. I think every time, and even a little bit with David Sinclair, I'm just like, you focus just on one part and it's really not the whole aspect. Like, and the brain is extremely complicated.
Well, yeah, it's like a car, right? So you can say, okay, the car is getting old and you're going to replace You're going to measure one part,
Yeah.
whether it's working, ignore the other 81 parts of it, and you're going to replace that part, make it better. Go for a checkup, job done. Doesn't make sense.
Yeah.
So, yeah, same concept.
So are you doing a lot of testing for patients? Do you see a lot of biomarker testing?
So, yeah, I think the most useful thing that needs to be done is to finish off these organ measurements,
Okay.
or like get those accurate. I think you can already do. So yeah, go back to like because I mean, obviously, brain is more important than hair for like clinical outcomes, right? Hair is important for obviously aesthetics, but yeah, it's not gonna kill you if you go bald. So yeah, like focusing on these, we already have quite good ways of measuring brain age, heart age, and I'd say arterial age as well. Like those three organs, they're quite easy to do. Because MRI was initially designed for the brain because it's a static organ, and it's really, really well studied for a long time. The arteries, we have, CT scans, which are really accurate for picking up plaque changes. And now MRI is getting to the point where it could be replacing some of the CT angiography as well. And you got ultrasound, and you got medical device-based markers. And there's lots of things you can do for arteries. And then the heart, you've got really good MRI models. You've got really good blood-based biomarkers as well, like troponins and NT-proBNP, BNP, some omic stuff as well, and also ultrasound, echocardiography. So those three organs, they're really important. And we have I can say we can measure the age quite accurately of all three of those organs now.
Do you do these tests on yourself regularly?
No, there's no point because I've done it like once, right?
Really?
Yeah, I've done it once, but there's no.
I think maybe I've done more testing than you.
Well, you haven't done any of these tests because this is like a horribly complex Excel sheet where you're typing in like echocardiography values for your heart and then like running regression, like hundreds of regression equations and then combining them to give an age of the heart. So yeah, I mean, only a few people in the world have done this test, for example, like with heart biological age via echocardiography. But it's not really useful in my case because I'm so young already. My organs are pretty young. I know how they're going to change. They're just going to age kind of a little bit slower than normal because I'm doing various, healthy lifestyle stuff. So it's not really this comes back to like why am I looking for someone with a huge budget to become this new guinea pig who's age 65 plus? Because 65 plus, we can show, it's a lot easier to show going from 75 to 70 than 32 in my case to 27. It's also clinically a lot more important as well because I'm not going to die anytime soon from aging. Yeah. Whereas if you're 65 plus, you're off the foot of the curve. You're on the exponential part for the all-cause mortality risk with the Gompertz equations. So it's much more clinically necessary to treat those people and show it can work in those people rather than young people.
What can people do to reverse their aging organs?
That's a complex one.
Everyone wants to know so that our organs don't age.
So I was frustrated at that question as well for like, 5-6 years again. And then systems thinking again, it's like, okay, there's so many variables that go into health.
I know.
How do we put it all into something that's, into something practical? So then I made the 1, 2, 3 protocol. So yeah, the easiest thing is do level 1.
What's level 1?
It's got 18 things in it now.
Okay.
It's been growing.
What is it? Sleep?
We can get an infographic up. There's a few. But yeah, there's like smoking cessation, exercise of a certain type.
Whole foods.
Yeah, alternative healthy eating index 2010 of a certain type. Sleep, B12, B9, vitamin D,
iron,
testosterone, Yeah, hemoglobin.
Goes on and on, I guess.
There's a lot in there. There's a lot in there. Yeah, ApoB.
There's not a one solution. Just take NAD.
You gotta do all those. NAD is not gonna help. I'm sorry.
Have you done your mitochondria test?
Which one?
Me screen?
No, no.
Oh, okay. Have you tested mitochondria? Is that something also you look at?
So yeah, mitochondrial aging. There's some cool urinary markers coming out for that, where you get the
Okay.
age
Okay.
of your like excreted mitochondria, so to say. It's very important because you get mitochondrial mutations, and you know it's easy to measure that in the blood and urine markers, but it's very hard to get those readings on the rest of the organs.
Yeah.
So you kind of have to
Each one.
guess. Yeah.
Yeah, I know. I think it's more complicated. I think there's the testing out there, but it's how accurate is it to say your mitochondria are healthy? I don't know.
Yeah,
if
you have
we're
to do
there
like
yet.
indirect markers most of the time,
But I
but you
do
can measure it directly in the white blood cells quite
Okay.
easily.
I think mitochondria are extremely important for longevity because it's your energy. It's your source of your body to have the energy to do everything, to, have a good immune system, to recover from anything. And your whole body needs the energy. So I think as you age, you have less and less energy. When you have disease, you're very fatigued, and that's when I feel like things are going downhill for people. So when you feel like you don't have good energy, that's a big sign. I feel that something's off, right? And I think that is pretty much a lot of the times due to the mitochondria, which with all the toxins in the world and all this happening, I feel like everyone's mitochondria is depleted. I don't know.
Have you heard of mitochondrial transplant?
No,
That could be a good solution. So,
no, I have really good energy.
oh yeah, I mean
But-
just in general, yeah.
Yeah.
Don't worry. I'm sure.
Yeah. Wait, so mitochondria transplant.
Yeah, yeah.
I'm so excited.
So I want it. So
Okay.
you know how we have like organ transplants?
Yeah.
Like organ transplants, you could fix aging if you can grow a young biocompatible organ
Yeah.
in the lab,
Yeah.
transplant it in.
Yeah.
You've solved aging in that organ in a lot of ways, right?
Yeah.
Problem is that's quite hard. That's a bit sci-fi right now because we're still getting there. You know, there's like basic stuff we can do in that for like quite a long time. You'd be surprised. Like, there's a lot of like Wake Forest Institute of Regenerative Medicine. Anthony Atala has some great work in that area, going on for decades now. But like the true kind of off the shelf, I can replace my heart and it's going to be fine. Kind of 3D bioprinted, bioreactor grown kind of stuff. That's still another 10 years away, I'd say. You know,
Okay.
5-10 years at least.
I feel like I saw that like 5 years
It's
ago.
always 5 years away, that kind
Okay.
of stuff. Yeah.
Okay.
So that's kind of still quite far
Just
away.
still the pig implant.
But the simpler way of a simpler concept, which is similar, is organelle transplants, right? Or cell transplants as well. So rather than doing the whole organ, you're doing like cells or organelles. That's a lot easier to do, and I'd say it's a lot safer.
that are made
you don't need
in a
surgery.
like petri dish, like,
Yeah.
Okay.
Essentially.
and then they would intravenously put these in the body, and it would go to the heart, it would go to the liver, it would go to the kidneys.
Yeah, depending on the targeting strategy.
Okay.
So there's one recent study.
Have they done this yet on a human?
I think the mitochondria guys, I think they've done a few mitochondrial transplants. Yeah, so they grow the mitochondria in a bioreactor, the young without like any copy number mutations or bad mutations or whatever, and then you transplant them in. So I think they've started doing that. It's quite a new company.
How are they starting this? Is this from stem cells? Is this from what's the
Yeah, so they're growing them somehow.
okay
They're growing them somehow. It might be it might be
donor? Yeah.
Yeah.
Interesting.
It might be like some kind of immortalized cell line that
Okay.
not a stem cell. It might be, yeah.
What are the most
we'll check.
advanced procedures out there that no one's heard of yet?
Well, yeah, related to that, I was going to say, because it just came out recently, November. So on the organelle kind of transplant topic, there's
Okay.
this. So and all these are things I want to try in mice to extend their lifespan, but in combinations with each other as well, because no one's doing. You can't cure aging with a single therapy. It's ridiculous.
Yeah.
So you have to do many combinations of therapies that make sense together, and no one's doing these combination mouse lifespan studies except myself and Aubrey de Grey as well, who's done the first one. So you need to think about all these things in combination and how that can work better than just doing individual things at once. So this other thing on this organelle kind of related topic is they I think it's a London company actually, but this is pretty crazy. It's not peer reviewed yet, so like the results may be half as good as what they show. But what they found potentially, because it's pre-peer review, was that white blood cells of a certain type, they interact with another type of white blood cell, then they join, and then the first white blood cell cuts off half its telomeres from all its chromosomes, packages it up with a few extra bits, a few extra rejuvenation based enzymes, stem cell related activation pathway enzymes, puts this in a vesicle and gives it to this other T cell, a CD4 T cell. And then so you got this glob of
White blood.
of telomeres. So
From a white blood cell?
from the antigen presenting cell, so
Okay.
one type of white blood cell, the
Okay.
APC, cuts off half its telomeres from all its chromosomes, roughly. I'm kind of estimating here because they haven't got the details in the paper, and then gives it to the CD4 T cell of a certain subset, and then the CD4 T cell sticks these telomeres onto its own telomeres to make it longer.
Lengthen them?
Yeah, to reverse them without using telomerase, which is normally the way that it's done. So it's like just gluing them on. And it makes sense because you can kind of, telomeres are very simple code. You just they just glue on to each other. It would make sense. So they glues them on and then specifically glues them on to the shortest telomeres within its chromosomes. So the most damaged chromosomes, they have the short telomeres that are starting to get double-strand DNA breaks potentially as well. It sticks on these telomere globs like Lego, literally like Lego. Lego is like the perfect analogy. It's like telomere Lego. So that happens, but this is only the first step, right? It's already pretty crazy. This
This is a procedure they're doing.
this is a natural process within your body
Okay.
which stops working with age.
Okay. So
So-
how do you induce this?
So that's the next step. So that's one benefit. So the APC is rejuvenating the
Telomeres?
telomeres of the CD4 T cell and helps it become a T memory cell potentially as well. That's why they're long lived. So they need a lot more telomere length to survive in that state, I think. But there's some left over. There's supernumerary telomeres, leftover telomeres from that glob that is sent over because it doesn't need all the telomeres that the APC has given to it. So then the telomeres that are left over, the cell divides, and then to one of the daughter cells, it gives a certain, especially prepared glob of these leftover telomeres, which it then secretes into the bloodstream, and then it goes to all the other organs in the body and lengthens their telomeres.
What?
But with aging, the various enzyme changes occur inside these CD4 T cells, and they can't do that last step. So they can't make the specially prepared glob of these extra telomeres, and then they can't spit it out and donate it to the rest of the body. So what they did in this study, which is still not, is going through peer review, remember, so it's pre-peer review. There may be mistakes in this whole kind of understanding. But what they did is they got rid of the buildup of fat. They had you get fatty CD4 T cells.
Oh.
you get a buildup of ceramides, the thing in CeraVe, the cream. You get
Okay.
like a
Yeah,
buildup
yeah,
of
yeah.
like a toxic buildup of that inside these CD4 T cells with aging. And
Interesting.
then
Okay,
that
we're getting
buildup,
down very scientific.
yeah, but it's like so cool, right? So like that builds up, but then they've given a new patented molecule to reverse that buildup of that, like the fattification of these CD4 T helper cells. And then the old mice were able to restart this process called telomere rivers and spit out these globs of telomere Lego. I guess I call it telomere Lego, and restart this whole process just by, I think they took out the immune cells, they bathed them in this drug to kind of like reactivate it, and then put them back into the mouse. And then they lived. It broke the mouse lifespan world record.
Wow.
But there are some dodgy parts in the paper because the results may be half as good as what they've stated. But even if they're half as good, it's still like a huge new mechanism of telomere biology that I haven't heard of before.
Who's behind this?
2 guys in London.
Interesting. Okay. No,
Yeah.
I've never heard of that. There's so many things. Like I interviewed Dr. Sheldon Jordan, like a top neurologist as well. And I know like the hypothalamus, like he has, I guess there was a study of mice and that you can, if you mess with the hypothalamus, the mice will age drastically.
Ooh.
And then if you put exosomes there with ultrasound, then it extends the mouse lifespan as well. Have you heard of that?
I think that was a disease model
Okay.
of, it wasn't wild type mice.
It wasn't
They're mice, but I think they were they're not like normal aging mice.
okay.
So they're kind of like a model of a certain brain condition, if I'm remembering correctly.
Okay.
Which is why I'm less interested in that. But those are useful as well. But like the gold standard mouse lifespan studies are like this one with the telomeres, where they take normal mice. They're not like genetically modified in any way to age faster than normal, and you start them around age 60 plus, human equivalent,
Okay.
with the therapy. And then you just let them age normally, and you make sure all their environmental conditions are really good and there's no confounding factors with like caloric intake or anything. And then you see what happens. So that's like the gold
Interesting.
standard way of doing aging studies.
I swear there's so many things that go into a study that people don't even think of. I mean, even
Yeah,
all the
even doctors.
pharmaceuticals were studying male mice compared to female.
Female.
And I'm like,
Exactly.
it's so different, right?
Mm-hmm.
So, what are also more of the top procedures? You are in the space of longevity talking to, do you ever work with David Sinclair by any chance? Have you ever been to his lab?
No, I haven't.
wanna
I've actually
go.
never been to Harvard.
Really?
Yeah.
Can we go?
Let's go.
I would love to go see his lab. What do you think of his like reversing eyesight?
Yeah, it's great. It's great
Yeah.
work. Yeah.
So I think he's saying. I mean, I've listened to some podcasts and Peter Diamandis, and it's like they believe that we can probably live to 200, or he even says infinity, or he wants to. So do you think we're going to get there eventually, and when?
Yeah. So
Do you talk to a lot of people in the space and saying, you know what, I do think this is something that we can reach in our lifetime?
thing is, you got to prove it statistically. And so, yeah, I came up with the concept of n equals 1 LEV, longevity escape velocity. So Aubrey coined the term longevity escape velocity, which is the time when your lifespan is increasing more than, or your health span and lifespan is increasing more than 1 year per year. So hence your lifespan is indefinite, your health span is indefinite. But no one thought about applying that concept to an individual as like a biomarker. Because what's your LEV? What's your velocity? Like what's your whatever,
Kind of the biological age, right? Like
Exactly. And then I was like, okay, how do I calculate this n equals 1 LEV?
yeah.
Once I came up with that concept. And to do that, you have to measure the age of all 82 organs and do it accurately, or at least, a sufficient amount of all-cause mortality risk from a subset of all of the ages out of those 82 organs.
Do you think, what do you think are the leading causes of aging? And I, as 2026, I think there's a lot of toxins, there's a lot of processed food, there's a lot of things kind of hindering us of aging or extending our longevity. So I don't know if it's honestly the best time. You have medication, we've had some things to extend our lifespan, but at the same time, I think we have a lot of toxins working against us. And that's going to be, I think, the hardest part about extending our longevity, maybe reaching to 200, when you have, cloud seeding and pesticides and all these toxins just surrounding us all the time. So what do you think? Is that one of the biggest factors for, extending our longevity?
Yeah. So, I mean, like, in theory, if you had different genes, right, you wouldn't age, or you'd have like built-in rejuvenation mechanisms inside you, or you'd age much, 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 much slower, as seen in like various other mammals, or the jellyfish that goes back and does potentially a OSK, Yamanaka factor kind of de-differentiation and kind of like makes its cells go back to like an embryonic like state, then comes back kind of fresh.
That's a jellyfish?
Yeah, the
I did not know
age
that.
reversing jellyfish. Yeah.
Oh.
Because we're like positively senescent, so we get our risk of dying increases with time from aging. And then there's animals which are negligibly senescent, which means your risk of dying from aging doesn't increase with time. And then there's negatively senescent organisms where, in theory, that's where your risk of dying decreases with time. Or you can interpret that as like you build in a kind of fun biological age reversal process into your genome. So yeah, this is like some niche examples, like certain types of jellyfish where they can do the like the David Sinclair eye thing, but it's built into their genome naturally, kind of an analogous way that they evolved. So yeah, the reason why we age, you can argue then, is because the reason we get any disease is because our genes, our chromosomes, don't have these built-in rejuvenation processes, which we see in other species.
So is that where we should be looking?
Hence, yeah, gene therapy, or even chromosomal therapy, or synthetic genome therapy, synthetic chromosome therapy. These are all very interesting concepts because, they're really powerful therapies that are then built into your body in the long term, and you can do a lot of things that would just not be possible with injections or intermittent therapies or medical devices or stem cells. Stem cells can be good as well because you can build in these artificial chromosomes or gene therapies into the stem cells, and then you inject them, and they can, if you do it right, they may engraft and become part of your body and spread out and overtake the old cells potentially as well.
Renew our organs.
Exactly.
Could be a start.
I
mean, I always think it's amazing, how we obviously have something in the bodies to reproduce a whole new human.
Which isn't the same age as us.
I know. It's
rejuvenated.
like,
Otherwise you just come out like age 40. Like, what the fuck?
And then you have a newborn. It's insane that we can do this. Honestly, it's very impressive to me that we're carrying it. Obviously, we're
all animals have that built-in simple rejuvenation process.
Yeah.
Right?
I think that's the key. We got to figure out how to get that all throughout our body. What do you think of animals? Like there's a shark that lives or like a turtle that lives.
Whales.
The whale. It's a specific whale. I think there's a shark too that lives a long time as well. It's like 300 years or something. Have you ever looked into that? Has that been something you're like, why do they live so long? Do you think it's an energy expenditure? Like,
yeah, I mean, not all of them are huge. You don't have to be the size of a whale you don't have to be a whale to live long.
It's not the mass.
It's not the sheer mass always, but there's a correlation, I think. And the things to take away from that is gene therapies again, because these guys got different genetics. They got like a lot of I think in like in the long-lived whales, they got a lot of like tumor suppressor extra copies. So like I think maybe we have like a certain amount of p53 expression, which helps reduce cancer. But then these whales have like a lot of p53.
Oh, interesting.
Don't quote me on that, but it's something like this concept of you have a lot more tumor suppressor genes, which then reduces your risk of cancer. So you don't get cancer even though you're like a much higher cellular number of cells. And even if you're a lower number of cells, you may, even if you're a human and had these genes, it may like solve a lot of cancer issues.
Do you think cancer and aging also is like our bodies start to get confused and our DNA gets kind of can't communicate correctly, the wrong genes get turned off or turned on, and that is also one of the leading causes of aging and disease?
Yeah. So there's like, there's Aubrey's 7 things. The Sen's 7 causes of aging.
Okay, I really need to read his books.
Yeah, it's good. So it's kind of like a more like a more simple and like in terms of physics, more comprehensive way of categorizing how aging occurs compared to the hallmarks of aging, which is based off Aubrey's work back in the early 2000s. The hallmarks of aging came out around 2013,
Okay.
building on Aubrey's work, Lopez-Otin, etc. So, but they're like 13 in the updated version. There's like 13 hallmarks of aging, but they're not like they still miss some things. I think in a way that that Aubrey thinks about. So I like to combine the two. I like to use both frameworks. But yeah, a lot of that stuff is related to DNA and epigenetics and hence cancer as well.
And do you ever study the misfolding of proteins?
Yeah, exactly.
Something. Okay.
Exactly, yeah.
Okay.
So
Another aspect.
another aspect, yeah. I mean, it's like
goes on and on. You're like, where do you want to put your attention to?
Exactly, yeah. What should we talk about? Well, yeah, going back to the environmental exposures, that's also another issue. So yeah, like why do we age? One is we could have these crazy good genes and chromosomes that means we're just like perfectly evolved and we can rejuvenate naturally within ourselves or whatever. That'd be like that's a reason why we age, because we've evolved to age. We don't have the right genes. But then the environmental stuff, yeah, it's pretty bad. Like part of my family, their whole family all died from pesticides.
Really?
Pesticide exposure, yeah.
In London?
No, outside London. But they all got cancers.
Wow.
Related from this pesticide exposure. So it shows how dangerous.
Yeah.
The American food system can be at least
so bad.
Yeah.
I always feel better when I leave, and I'm like, I don't know if it's worth living here just for my longevity, for like my health. Honestly, like
it is a minefield.
It is something that like I try to only do organic, and now regenerative farms. I don't even trust Whole Foods, and but it's now I feel like I isolate myself from just having like enjoying some freedom. Like when if I'm in Greece or something, I know the food system's a lot better. I can have more freedom going out and enjoy myself. But here, I to be healthy, you have to be very strict.
Yeah, it's such like a cognitive burden here as well. It's like UK, I can just go anywhere, and it's like I know I'm not gonna get fucked. I'm gonna be like, I'm gonna be okay. Even if I eat junk food, I'm gonna be okay. But here it's like it's so artificial.
It's not junk food. It's like chemical food.
It's like chemical, yeah. Like I got this like organic whole grain bread.
Here?
Yeah. But then like I didn't realize they had like 10 grams of sugar per slice added in. Who does that? No one does that anywhere else in the world. So then I ate it and I got really sick. I had like, I was 48 hours. Yeah. Just poisoned.
I don't even trust organic. It's just, I don't know. It's a whole system that I think a lot of people are fighting. And Honestly, I feel like it's affecting your mental health. Like a lot of people I don't think are healthy and they don't even know how it feels to be healthy here because they've been toxic their whole life and they're just now used to it.
Mm,
But like and I used to and I felt like that probably my whole life. I felt brain fog, exhaustion, like all these things and then when I started biohacking and taking being proactive with my own health, that's when I was like, wait, this is how I'm supposed to feel. I feel probably 98% of people in the US don't know how it feels to like feel
mm.
healthy, truly healthy, you
And
know?
like Miami Beach and Brickell and downtown, it's like it's the
Not,
1%, right?
I
It's the 1%.
know, and you leave. So, okay, while we're here though, because it's like I have you here, let's talk about gene therapy. Let's talk about maybe some plasma exchange, follistatin, Clotho, peptides. What else is there? Like, I want to talk about the like advanced longevity protocols that are happening.
Okay, okay.
Gene
Where do you want to
therapy.
start?
Let's talk about follistatin. And what do you think is the future for gene therapy?
Yeah, so I'm flying to Bahamas soon
Okay.
to see for like a biotech meeting with the government to see if we can run gene therapy clinical trials there
Okay.
in a safe and effective way. That's
It's the one they are doing gene therapy. I think they're doing mini circle.
yeah. So mini circle. So we'll talk about follistatin.
Okay.
So yeah, follistatin, for anyone listening, it is a naturally occurring gene that you have. So it's already in your body, but it's at a certain level where it's not too high because we've evolved. Again, this goes back to like the evolution point, right? Our genes aren't evolved to live past. There's not evolutionary pressure to live past age 40, roughly.
You
So
think 40?
probably, or 30, yeah, or 30, 40. You look at the long term, like average lifespan once you get past childhood mortality. It's something like 30, 40, isn't it? Before
Yeah, quick
things
question. Like, what do
things
you
go down.
think we were meant to live to as
Um,
humans?
what? Uh, if
Without
you get
without
past childhood
toxins.
mortality,
Without toxins, without anything happening, like what do you think
oh,
the
okay, I
body
see.
how long do you think the body is supposed to like live for?
I think we've evolved roughly like, I think, I mean, it's probably 80, probably 80.
I think so. Okay,
It's,
okay.
yeah, I mean, probably 80, I'd say.
I think the oldest is a woman is like 120
Yeah,
something
but it's like so many standard
factors.
deviations away from 80. Like,
Yeah,
it's
yeah.
like an outlier, but
Okay.
whether
False.
that's, kind of, it's not relevant for most people. But yeah, so follistatin, we have this gene, but it's very low level because what it does, if you turn up follistatin, it's going to turn off activin A, activin B, myostatin, GDF11, and some other stuff related to the TGF beta family of molecules. So a lot of those things, they prevent you from gaining muscle. And that's because we don't want too much muscle when we're evolving because it requires more caloric maintenance potentially as well, or at least protein, maybe more protein maintenance. So that's not a good evolutionary strategy. If you're walking around with like 70 kilograms of muscle as like a 120 kilogram person, although the Denisovians, which are kind of like there's Neanderthals, humans, Denisovians, like another kind of offshooted species. Apparently they probably had very high follistatin levels. Maybe that's why they died out. I don't know. Maybe they're too jacked. But apparently they were like 100 kilograms.
Takes more energy,
And
I
and they
guess.
were super muscly, like
Yeah.
very, very muscly. So it'd be interesting to see what their follistatin levels would be predicted to be. But yeah, it seems to be that we didn't evolve follistatin to be super high because of caloric pressure or protein pressure. But we have found that giving follistatin in preclinical studies in mice can do a lot of good things and potentially lengthen health span and lifespan a lot as well. Because you get this age-related muscle loss, sarcopenia, which may not respond to hormone replacement therapy, to strength training, or to protein intake as well. So you need all those things may not be sufficient for a lot of people, and especially when you're in a kind of situation where you can't go to the gym, or you can't eat enough protein, because you're not mobile. Say you have cancer, right? And you're doing chemotherapy, you're not going to be going to the gym.
Yeah.
So, yeah, it's like there's a lot of situations where that occurs, especially as you get older. You know, your immune system starts going down. You got infections all the time, pneumonia, heart disease, heart failure, whatever. You know, osteoporosis, hip fractures, everything. Lots of different causes that lead to frailty and sarcopenia and loss of this muscle volume and basic ability to do the activities of daily living, is the technical term. So follistatin comes in. You can upregulate this gene in your body by giving a gene therapy that gives you an extra copy of this gene outside your chromosomes, but inside the nucleus of your cell. So it's like floating around as a little plasmid or a little circle. And then this is expressed inside certain areas of the body where you want it to be expressed locally, in a certain way as well, in a way that is safe. And various other complex things we won't talk about because it's like gene therapy design, which is a bit too complex. So there's different ways of doing that, of putting this gene inside your cell, the follistatin gene, to get these muscle benefits. And also other benefits we've seen preclinically as well is reduction of scarring throughout all the organs in the body, or fibrosis. Very common way, it's a fundamental aging pathology, fibrosis, like across many different organs, like the lungs, the heart, the kidneys, the brain, not the brain as much, but like lungs, heart, kidneys, for example, pancreas as well. You get a lot of scar tissue of different ways built up, or hypertrophic tissue, too much of the wrong type of tissue. And then this just stops the organs functioning. It's not good. In the muscles as well, actually. Very dangerous when you get it in the muscles. So follistatin may reduce that, the scar tissue built up throughout the whole body, and also the calcification process, another fundamental kind of aging process, this ectopic or unwanted calcification, where calcium is being deposited in places where it shouldn't be, like in the arterial walls, which is a protective mechanism somewhat as well, but also in many parts of other organs throughout the body, including the pineal gland, which gets almost completely calcified. And you can see it when I do my MRI scans and measure the age of the pineal gland.
Interesting. I know. I think a lot of people don't think about detoxifying the pineal gland.
It doesn't work because they haven't measured it accurately. Like no one does. You need an MRI.
What do you do for the pineal gland?
I've got various ideas, but you need to measure it first. So
Okay.
You need to measure it first. That's the thing to take away. So, by your pineal gland, but we have to measure the calcification first to show that we are decalcifying it rather than like all this woo-woo
random
crap. Yeah, unfortunately. So, yeah, going back to the follistatin, it can get the muscle gain, the functional improvement as well, and it's not just like you get bigger and nothing happens strength-wise, and then you get the potential reduction in whole body fibrosis and whole body calcification processes chronically for many years. Because once you put this therapy inside you, if you do it in the right way, it stays inside you from a single injection for many years, or 10 years or 20 years, because it's going inside the muscle cells particularly, or certain organs that don't divide and wash out the therapy. So there's one gene therapy you can do which uses the herpes virus, HSV1, but as everyone knows, you can get herpes again and again. You don't build immunity to it because the HSV virus is a way of avoiding the immune system and reinfecting you. And that's why it's a good gene therapy virus for the skin. So they give a gene therapy to the skin for a certain rare condition, this one is, and you can redose it without getting immunity because you're using the herpes virus, but you have to redose it because the skin is always turning over and shedding the cells, as you know, and washing out the gene therapy. So it's an issue. You have to redose your gene therapy and use a certain virus to make that gene therapy go in and do it that way. But with muscle, it's a lot easier because you're not like shedding your muscle cells every 90 days like the skin. It's really like they kind of stay the same number roughly, and it doesn't really wash out at all compared to the skin, for sure. So depending on your type of gene therapy, where you target it, where you want it to live inside your body, it can stay there for a few days if it's a really high turnover target cell type, or decades if it's the opposite. So in this case, we're targeting the follistatin to the muscles, the skeletal muscle, both fast and slow twitch, and this may give you these huge gains in muscle mass and benefits in calcification and scar tissue buildup as well.
Is there, so I thought a lot of these gene therapies, which is already happening, it only lasts for about a year or so. I didn't know if there was clinical trials or something.
They did their own study. They did their own study and showed it didn't work.
Oh my
They
gosh.
showed it didn't work for muscle gain or fat loss,
I
which
know a
is
lot of people have been doing it.
yeah, but
Even Bryan Johnson did it.
yeah, because they just
Did
out
you
of
see a
curiosity.
muscle mass increase after he did it? Wow.
No, they did a trial on 100 people, which didn't have any. They're not like bodybuilders already. Like they're normal people. These people, because if you're a bodybuilder, it's gonna be harder to gain muscle because
Yeah.
a lot of bodybuilders are interested in this as
Already.
well. But yeah, they showed it didn't gain in normal people. No change, and they're using a very small amount of DNA, and they're also it's- they're not really. It's not copying how the preclinical data works. They're just like putting a bit of this follistatin in your fat or in one muscle, and then it kind of just secretes out from there. It's not really when you use a virus to deliver the gene therapy, which is what all the preclinical data shows, and the existing clinical trials for rare muscle diseases where they use follistatin in a virus. They're using a virus to deliver the gene therapy to all of your muscles around your body. And then it's coming out of your muscles locally and acting locally around all the parts of the muscles across the whole body.
Oh, wow. Okay.
So
So,
it's very different.
yeah.
There's a lot of, and then if you underdose it, or you use the wrong promoter, like part of the thing that chooses which cell types your gene therapy turns on in, or you use the wrong silencer or enhancer or microRNAs or whatever, then it's not going to work. So a few companies have tried this, and there's no evidence it does anything, like BioViva, seemingly, and Minicircle, and yeah, no one is no. A lot of companies have failed, even though they're using gene therapy in a non-viral form or in a lipid nanoparticle form or in a viral form. They've all failed. But there's a few, if you get everything right, it can work. You need to get all these ingredients in the recipe right. That's the way of thinking about it.
Has this been for this procedure that you're doing has it been done on a human yet?
Yeah.
Okay.
So I've got a colleague at Triple Helix,
Okay.
Dr. Patrick Sewell. He has some interesting data.
Have you haven't done this on yourself?
No, not yet.
Next year I'm gonna see you're gonna be like all muscular. The gene therapy also, I'm glad I didn't go and do this because I was invited to go down to the Bahamas and do the follistatin. I am kind of the biohacker guinea pig, so I don't really do everything to protocol, but
No,
but
stuff.
it has helped me. So being my own guinea pig, and then there's also Clotho coming out as well. So do you think that's going to be extremely beneficial? And I've tested my Clotho. Is it easy to test for, and how accurate is the testing?
Yeah, it's tricky. So again, like going back to follistatin, Minicircle, they raised blood follistatin levels 2 or 3 times for a year, like sustained, 6, 9, 12 months, but it didn't do anything. So it's not just about measuring your blood level, right? Because that's just one thing in your body. A lot of proteins and molecules, they don't work that way. They're only really, you need the tissue level of a specific type. So you have 82 organs,
I did
right?
not think of this.
Think of it this way: 82 organs. You have 82 different levels of every single molecule that you can imagine in your body, and the blood level is just one of them. And within the blood, there's many different types as well. And within the organs, there's many different types because each cell type is going to have different levels potentially,
Yeah.
either outside it, just next to the cell outside, or inside the cell,
What
or
about
even in different
Clotho?
parts of the cell.
Is Clotho
So it's complicated. It's
is
fucking
Clotho in all the
complicated.
organs, or just
Clotho-
that for just the blood?
Clotho, I'm not an expert on yet, but
Okay.
basically there's kidney Clotho. So when your kidneys age, you reduce kidney
Clotho.
produced Clotho, and then there's brain sourced Clotho. I think there's a few other sources as well, but I think these are two of the main sources you have to think about. And so that's why you have to think about when you're doing a Clotho therapy. Are you recapitulating the natural young environment of your Clotho system in your body, right? So is there evidence that your mRNA or protein or peptide or gene therapy for Clotho is doing that? Like, is it restoring brain levels? Is it restoring similar levels to what the kidney is producing in the same kind of way? Is it going to the same target tissue distribution? Have you done like analysis on, what are tissue levels of this, and is that being restored? Like, are you tracking your protein with like a fluorescent or radio labeled marker to see on biodistribution?
Way more complicated than I thought.
and yeah, like so both these have mouse lifespan studies showing roughly 20-30% increase in health span and lifespan, follistatin and Clotho separately. But with the case of Clotho, it's a knockout study, I think. So it's showing that if you don't have Clotho, you're living 30% shorter. And if you have, from birth, if you have an extra amount of Clotho, that could double the gene, I think then you live longer. Something like that. So basically, there's a problem with the Clotho mouse lifespan study in that these are started from age 0. So we have no idea if it works the same in adults. All we know is that, Clotho is very important and it declines a lot with age, but we don't yet have good evidence that there's a certain gene therapy that's going to reproduce the benefits of Clotho across all the different organs in the right way to give that into humans.
Okay.
In old humans already. Because, if you give it in mice at age 0, that's like, you can't compare those studies because you may have to have this change in your puberty for it to give any benefit in the rest of your life. So all the benefit may be from as you're growing up, as you're seeing with like other mouse lifespan studies, like looking at growth hormone or dwarf mice and so on. So Clotho is cool. You can measure your blood level for around $200 now if you go to various places, like Gin Affinity, I think, has a test, and
which I've done.
Yeah, and
My levels are good, but I don't know.
What was your level?
I think it was like 7 or something.
I think that's a different number. If it was 7, you're
No, it was good. Like, I was a little surprised. So.
Maybe different units.
Okay, I'll have to look it up.
Okay.
I'll show you.
I think it's normally like much higher number.
Okay.
In my units.
It was a good range.
Okay,
so
was a good
whatever
range.
the number was.
So
what about injections? Because I know there's some companies that are almost like peptides.
Same with follistatin, right?
Yeah,
I mean,
follistatin.
you can get any protein you want, really, and inject it.
Okay. So it does work?
You can ask an AI how to make it, where to make it in a research use only case, in a lab. Anything patented, you can make it in the lab for research use only. Okay. And then you can inject it in yourself.
Okay.
And if it's a short protein, it's called a peptide. And then you can do whatever you want. But does this do anything? We have now with AI, you can actually do that. Yeah, no one could do that before. They wouldn't really understand that's a thing. But now AI can tell you exactly how to do that.
Okay.
But we have no evidence.
Okay.
That
so we don't know if it is helping.
It could be.
Maybe some people might feel more muscular. I don't know with the injections, but I think you have to do it daily.
I know, but placebo effects. Okay, you tell people. There's loads of placebo studies on testosterone, right? You tell them they're taking testosterone, and it's pure placebo, right? These are saline injections, or saline patches, or saline gel, right? Just like nothing in there, and then they gain like insane muscle mass and insane functional strength gains because of placebo effects. So, I mean, that would actually help because gaining muscle and strength gains in those cases would be good. So the placebo does work.
Okay.
In some cases, it does work. It's not going to work for like cancer, but when it's like a lifestyle behavioral thing, placebo can be a useful strategy. Okay. Not that I've ever used it actually, but the placebo effect can be so strong that you just these subjective markers, like you have to use objective markers. Like you can't placebo your thymic age on 3T Dixon MRI and SJT Rex qPCR. You can't placebo that. It's impossible.
Yeah.
But you can placebo, I feel better.
That's true. And I mean, it's a new thing. So I don't think, just like with Clotho, I feel like there's just not, it's very new. And so we're just starting to look into this and
Yeah.
see how
But the
this
problem is,
is.
like, Clotho, we already know. I mean, not Clotho, follistatin, we already know with minicircle, you have a high blood level of follistatin, like 3 times the normal range, doesn't do anything.
Yeah. Yeah.
So doing the peptide is going to do fuck all. It's going to do even less. We already know you have like a permanent peptide being released inside you in the wrong way.
Yeah.
We already know. So follistatin definitely does not work.
Yeah.
Doing these 2-hour
If you,
half-life injections anyway. You might as well take creatine. Like creatine has some follistatin like
Huh.
effect anyway, or
Oh, nice.
myostatin
Yeah.
probably. So just do that.
You just do the
Much.
creatine.
Then the Clotho's a little different because you can get some types of Clotho, maybe mRNAs or modified peptides, which may have a longer half-life and may make it more realistic. I think follistatin is very hard to do that, so that's why it hasn't really been successful. But yeah, you can, in theory, you can modify these. You can modify any peptide to try and make the half-life longer, or modify an mRNA to make the half-life very long.
Oh, interesting.
Because that's kind of like how a lot of drugs work, like cholesterol drugs. Recently with like inclisiran for spool inhibitory
What are
RNA.
What are your thoughts on GLPs?
Yeah, they can be good. They can be good.
GLP 1, 2, or 3?
I don't know. Like you only need them if you as like. Yeah, I think there's like in like a longevity
I just listened to
perspective.
Bryan Johnson. He thinks microdosing. I think red at GLP 3 is there's so many benefits to.
Yeah, I don't know. It's kind of
I like it.
there's so yeah.
I think
you have
Rada's
to, you have to use a
amazing.
guideline. You have to use a
Yeah.
guideline because you can lose muscle mass.
Yeah.
You
I
can
think
lose bone mass.
I think so many people are not testing. You need to be testing, doing a full blood test function over 100 biomarkers, making sure. I feel so many people are just guessing. They're taking supplements even and doing all these things. And it's like, you could be harming yourself by even just with supplementation. So it's like a lot of people just want to pay for the supplementation, the peptides, whatever, and not test. And I'm like, it's the craziest thing to me because I'm like, you should be checking all your biomarkers first, wasting money, but also you could be harming yourself. So even just simple supplements, and even your diet, like maybe you need to like be eating more seaweed for iodine, or maybe
Yeah, probably.
I mean,
Iodine deficiency.
me too, and
Nice. My level was so low.
And
So
one of
low.
these biomarkers can change everything for you, your metabolism. So
Yeah,
like start
exactly.
there with these testings first because you want to, lose weight. It could just be iodine or it could be iron or something,
Right. Right.
So I do think a lot of people are jumping to peptides and jumping to supplementation without doing the testing first. So I'm completely for fully doing all the
No,
testing.
they're great. They're great in many types of obesity, but it's
Yeah.
kind of like what do you? I mean, yeah, like I think you gotta know your like zone 2 strategies. You gotta know your diet strategies.
Yeah.
As well, like doing
And not using it as an excuse for unhealthy habits. I think a lot of people are like, I'm going to eat a cheeseburger or processed crappy food that one time I'm going to eat today.
Yeah, exactly. Yeah, I mean, like the best diet is the one that optimizes your biomarkers
Yeah.
for you. That's the answer for diet, right?
Yeah.
As much as you can prove that, because a lot of it you can't. People don't understand biomarkers, so they won't know what to measure or how to interpret it. But yeah, with GLPs, I think they can be good, but you've got to think about muscle mass prevention, muscle mass loss prevention, bone mass loss prevention, gastric paresis prevention, pancreatitis prevention, and other side effects as well. So there's quite a few, and too much fat loss as well. Like you can get a facial gaunting, or it just makes you so old, and you got to regrow that fat, which can be tricky or very expensive with all the fat stimulators. So yeah, I don't know if Ariana Grande did she have
I think so.
Yeah, it's like really like, aged her facial fat.
I think I think most people are probably on it.
So that's the thing. Like you need a guideline. So no guideline, no therapy. Same with tests as well. Like you don't understand how to do a test. When in a complex test, it's like no guideline, no test as well. Some tests can be very complicated to do. And
I think it's just a lot of people don't want to spend the time or the money to do all that because it does
Yeah.
take time, it does take money to kind of do all this research and the biomarker testing. So they're just like, how do I lose weight? Let me just order this injection.
Just take GLP.
What are your thoughts on stem cells? That's a big topic too now, where a lot of people are doing stem cells to either recover from an injury or, there's so many different procedures for them, and there's a lot of different stem cells out there. So
A lot of scam cells. Yeah.
yeah. And so what do people do when they think, okay, I want to, maybe look into stem cells?
So
What are good stem cells? Where do you
there's
go?
so many facts out there. I reported one recently to various governments.
Wow.
Yeah, watch out.
It is. It's a scary situation. There's donor base. There's so many different types of stem cells, fat, this, and then they replicate them from one donor over and over and over. It gets very complicated. A lot of people just don't even do the research and they're like, I need to go down south somewhere and go get stem cells for my knee injury or something. So
Yeah.
how do people know it's good stem cells, and what do you think are the best stem cells out there?
So one problem with stem cells can be good for like lungs and joints. They're kind of the easiest clinical scenarios, I'd say. They can work for a lot of organs, but it's all kind of still in, you can get you can get specialized types of stem cells injected into your coronary arteries or your pericardium or your substantia nigra in your brain for dopaminergic neurons in Parkinson's. But these are all very hard and complex to access. So in terms of there's kind of two things. There's like the complex stuff, which is in very specialist research and very dangerous as well. And then there's like the simple stem cell stuff, which is more like lungs and joints.
Intravenously. So you're
Yeah,
kind of
intravenous
saying,
or intra joint.
okay.
Even with that though, there's loads of scammers.
Yeah.
I mean, there's few scammers Scammers can't really do intracranial injections.
Yeah.
So there's
Makes
or like
sense.
intracardiac injections or intraarterial injections. They're normally like that's too complicated for them normally. They want easy money, right?
Yeah.
So, and they don't want to kill the patient because that's actually very dangerous, this kind of interventional radiology. So they like to stick to the easy stuff, which is like joints, which is often done without the correct imaging guidance. So they're doing it without the correct ultrasound or the correct fluoroscopy, X-ray guidance. That's a common mistake you should watch out for. Basically, I've got a checklist. This is why I have longevity school, because there's so many. One of the modules is just the scams module.
Really? In your
Literally,
longevity school?
The scams module, yeah. So it's like various checklists for different kind of everything you can imagine. Is like skincare. Like, what are all the scams? Why are they scams?
In
Like
your longevity school?
Yeah, like supplements. What are all the scams? Why are they scams? Stem cells. What's the checklist you need to do to check that this is
so cool.
This is actually a stem cell. It's actually alive, and it's not dying, and it's actually not going to cause graft versus host disease. That you're not going to get HIV or hepatitis. All these things. But also, yeah, that's like kind of the scams aspect of a lot of this. A lot of these people these days. Mini cycle as well, arguably, if they're promoting it for muscle mass. I mean, I don't know. Yeah, I don't know. Doesn't why not do? Maybe they're doing good research. I don't know. The branding isn't good, right? The perception isn't good.
Yeah, yeah.
Negative results are great, but the PR, the PR is not good. It's very bad. It's not professional. So, yeah, problem with stem cells is you often don't need stem cells, right?
Okay, a lot of people say stem cells have healed them.
I know, but they're expensive, right?
Yeah.
And there's lots of things you can do before, people can't afford them, right?
Yeah.
Maybe you can jump to them if your orthopedic doctor recommends it, But like even then, you can't because there's not very few insurances cover stem cell therapy.
Yeah.
A lot more insurances, and it's just cheaper anyway for self-pay. It's like PRP, shockwave therapy, laser therapy, specific types of specialist physiotherapy. There's many other things which have very good evidence for many musculoskeletal injuries. This is not talking about the lungs, just like joints at this point. But there's many other things which are very cheap and covered by insurance, so often very, very cheap, which aren't stem cells. So you shouldn't jump the gun on stem cells in a lot of cases. You should
What
look
if you have a
into
lot of money and you're like,
if you have a lot of money, yeah, you could jump it. But
do you think? But what makes good?
then you have to go through the hassle of like verifying your stem cell sources.
Okay, so
There's
how do you
admin to
do
it.
that? Because that is the ultimate. Like, what are the best stem cells? Where are they coming from? The placenta, the umbilical cord, or do you think only just from your own?
Yeah, so the most randomized, there's hundreds of randomized control trials across all these modalities, all these types of stem cells. But I think the most RCTs are done on autologous. So where you're taking it from your own bone marrow, typically. Those show the best outcomes. They have in terms of evidence, they have the best outcomes evidence for many types of joint injuries.
Okay.
But again, you have to think about the specific joint injury. You have to think about whether it's multiple injuries within the joint, where you might need multiple types of therapy as well. Like stem cells just won't work for certain injuries. PRP won't work for certain injuries. Shockwave won't work for certain injuries. Physiotherapy won't work for certain injuries. Sometimes you just need surgery combined with all these other things or whatever. So you can't just summarize the whole of musculoskeletal medicine in do umbilical stem cells. It's not how it works.
I know like Dr. Joy, and she looks amazing. She does a lot of stem cells and everything in California, but she's all about it. But she's in her 50s. She looks really good. And she's all about the stem cells. And I hear a lot of people, and this is donor based, like umbilical,
Yeah.
yeah, that people just, it's, and I'm sure, like Dr. Jonathan Coe of like, I think he's a fan of the Muse stem cells. And so
The mysterious Muse stem cells.
I know.
Mysterious Muse stem cells.
But I do hear good benefits. So it's,
I know, but going back to placebo.
You think I don't
The
know.
placebo is very powerful unless you have
Because people will struggle with this their whole life, and like, or a back injury, like the Kardashians. I don't know. They inject it into the spine or something from back injuries. Did you see that?
Shoulder, yeah. I mean, you need MRI evidence to show an objective endpoint, right?
Yeah.
Like those kind of symptoms where it's not like if it's spinal cord injury, right, or like paralysis,
Yeah.
or extreme changes, like huge changes in WOMAC score in the knee or whatever, like your DASH score for shoulder or whatever. If it's a huge change and multiple time points, this is like n=1, you're trying to prove it n=1, right? You have to think about case reports, peer-reviewed criteria, what would be required to get this peer-reviewed and published as a case report.
Yeah.
Like these doctors aren't doing that.
I know. I guess it just comes to
It's
a
like,
point
oh, my
too,
shoulder
where it's
feels
like,
better. Everyone should take Muse cells. What are Muse cells? I don't know.
I mean, I think people are coming to the point where it's like, do I want to wait 10 years for the research to come out or do something
Or
now?
they could just do a bit of PRP and shockwave.
I've heard many times where PRP doesn't work for people.
Because they didn't follow my PRP checklist, and they're using fake PRP. Well, not most, but like a lot of PRP is fake. A lot of stem cells are fake.
Yeah, yeah.
Shockwave's harder to fake. That's quite
That would make sense.
I mean, you can't fake a shockwave. You can feel that.
Yeah, I just,
Shockwave fixed my shoulder, right?
Yeah, maybe I need to do some
Cost
of
me
that. and do
like
some
100 quid.
release. That what?
Cost me 100 quid.
Okay.
No blood draw.
Yeah.
No.
Yeah,
it wouldn't even work.
I
Stem cells PRP wouldn't even work for like so many. Yeah, it wouldn't for my shoulder injury. It would be. It's not indicated.
Yeah, for some things you definitely won't. I mean, if there's a cheaper version, most
If
people.
you're showing objective MRI evidence of like T2 map, 3 Tesla, cartilage regrowth, cardiogram sequence or whatever, multiple time points before and after, that's, borderline case report. Or, you have to think about, is this peer-reviewed case report worthy? Otherwise, it's just junk. It's junk science. It's not science. It's just...
What if money was no issue?
You do the same as you do in MRI.
Well, yeah.
MRIs are expensive.
No.
This is why no one does it as well. Like,
okay, say money was an issue and I wanted to go do stem cells. And I want to...
Okay, so you got a billion.
Yes.
Okay.
I have $1 billion and I want to go do stem cells. What would be the best stem cells to do? No, I want to do it for longevity.
That's what I mean. Like, markers, longevity.
You won't give me an answer.
No, so I want to measure your organs first, right? We need some indication. We can't just do it and not have an outcome that we
just
don't know
do an
what's
intravenous into the blood stem cells.
You just want to see what happens. Is that what you mean?
Yeah. Shouldn't my stem cells go to a certain area where they need...
Yeah, they'll get stuck in the lungs and then die there.
Die off.
Yeah. And that's only if they're alive in the first place, because most people, they do a very... most providers, they won't tell you whether your cells are alive. So you might be getting dead cells, which then causes like a necrobiosis kind of process, which can be maybe part of the benefit, but it's also like... It's not really what you're going for a lot of the time. So you're getting these dead cells, or they're alive, but they just do something. They do a very simple live dead stain to check how many are alive. That doesn't tell you if they're going to die in the next 10 seconds, or if they're going to die in the next day or week, or divide.
Yeah.
So these assays, they're like tests. Assay means test. To check whether they're alive, which is very important because if they're not alive, they're not going to be as good in a lot of cases with stem cell biology. There are some benefits of having dead cells injected inside you, but that's not normally the intended method. So these providers, they're not really testing functionally whether these cells are alive and dividing and performing normally. Like with MSCs or whatever, they're not even doing a live dead stain normally. And if they do any kind of test to see whether they are alive, they're doing a live dead stain. And that doesn't really mean much about the cell health at all. You want healthy, fully functional, dividing, reacting cells, clarification, before doing these kind of injections. That's not an issue when you're doing autologous, your own cells, because they're alive and they're minimally processed typically, or cultured a few times if you're going to like Cayman Islands. So it's not really a big deal there, but like even then you should do the test if you're doing autologously. But especially with these allergenic cells, umbilical, placental, whatever, Swedish medical students, then you want these kind of clarification.
Do you think they need to freeze them right away? Is it the shelf life?
Other things as well, like how many freeze-thaw cycles have they done? How many? What's the culture medium? How many passages have they done?
Yeah.
Etc. These are...
Do you think these are questions that people should ask if they are getting stem cells?
Yeah.
Is there a checklist?
Is it GMP? That's like a basic one. Good manufacturing practice.
Okay.
So these are all the issues with allogeneic cells.
Yeah.
A lot of these are solved when you're doing autologous.
You have no idea when you're going and getting them. It's like, where are these from? What was the
procedure?
Placebo. Yeah. Maybe. I don't know.
A lot of the time. Yeah. It's a random guy in Mexico or random chiropractor. Or who doesn't know what a culture assay is, or like doesn't know about HLA-G.
Yeah, it's a.
No one knows about HLA-G.
It's a risk factor, I guess, that you have to take, that you're gonna take when taking.
Or maybe you don't even know the gender of your cells. That's important.
Is there a risk factor to it then? Besides the money, wasting money.
Well, yeah, it depends where you inject them. If it's in joints, you can get like very serious side effects if the joint injection goes wrong, like joint infection, or nerve damage, arterial damage, especially in the spine, very risky.
Yeah.
So people doing ultrasound guidance for spinal injections and no fluoroscopy, it's like criminal.
Yeah.
Should be criminal charges.
Yeah.
To be honest, very common, very dangerous.
Wow. What about
Yeah.
exosomes? Are you a fan of exosomes? Do you think
Exosomes.
they last longer?
Which ones? That's like, are you a fan of mitochondria?
The ones I get from a medical spa. No, just
Oh,
I'm
those
joking.
ones. I mean, I tried them all.
You
I
have
tried them all. Yeah, I got
on
a
your
skin clinic.
skin?
I got a skin clinic as well.
Yeah, yeah, I know.
Didn't
You're kind of everywhere.
I got? I don't know. Exosomes. Um, I've seen a few people get like necrosis
What's
when they
necrosis?
inject them.
Oh, yeah.
Necrosis, like the tissue just rots and like you get like fat loss, like tissue
Wow.
loss when they're injected. So yeah, that's why it's illegal to inject them
Mm.
because they're not studied.
What about salmon DNA?
Yeah, the
Have you done
PDRNs.
that
Yeah, I've done it. I've done it.
on your face?
I actually cried. And I more than I cried and I screamed.
Do you
Screamed.
think it's beneficial?
I saw no benefits because you need at least 4 sessions.
Okay.
3 sessions, 6 sessions ideally. It was just too painful because the pH is very acidic, right? So I do it. I'm good at numbing people, right? So I know how to numb with my amla cream. You do you want a certain, there's like an optimal time for each kind of tissue in the body for lidocaine, and for skin, it can be like you want to do quite a long time, thick layer, mask over the top to make it go in. So I was numbed up, couldn't feel anything, but it doesn't matter because like that doesn't stop the pH acid burn pain when it's injected.
I've
So
done it a few times. It didn't.
I found it. I was literally in so much pain. Probably the most painful thing I've ever had. Yeah. So I had
Wow.
like 20 injections each side under my eyes
Oh, wow.
for it.
That's kind of a lot, I guess.
Yeah.
Okay.
I think a better solution for some of the under eye stuff that may be a better solution is maxillary expansion. So you are like a mini screw assisted rapid palatal expansion or slow palatal expansion or dome procedure. These are different names for maxillary expansion, where you have the orthodontic device inserted into the roof of your palate, and then it expands it and fixes all your nasal issues and occlusion issues often as well. Very common in orthodontistry. But that actually can, especially in someone like me with a small maxilla, small palate, it can help with the bone support with the zygomatic under the eye, and that's how you getting rid of dark circles and periocular wrinkles.
Oh, interesting.
Skeletal.
What about mouth tape? Is that something that you
That's
think
good as well.
helps?
That's good. Mouth tape's
Kind
good.
of like readjust.
But you're not fixing the root cause if you're doing mouth tape, unless it's purely behavioral.
Okay.
If you're like mouth breathing,
Yeah.
then maybe it's because your nose needs
Is-
surgery.
Yeah. I think a lot of people have issues. I always hear people like breathing weird. Like I can hear it in the background. I'm like, you're struggling there. I can imagine how that's affecting. And then it's your whole nasal passageway.
Yeah.
It just affects everything.
I just had a
So-
nose surgery actually
You just did?
2 weeks ago. Yeah.
What did they do?
I got a bilateral cartilage radiofrequency tightening for nasal valve collapse, and then I got an out fracture of my inferior turbinates with a coblation of radiofrequency at the same time for shrinking.
I have no idea what that means.
Basically, it means
What was
They-
wrong and what did they do?
so like there's this thing called a Cottle Test. So you can do it. So you pull here, either side, just on your bit more central on the cheeks, like lateral to the nose. Yeah, pull out, and if you can you breathe better through your nose as you're pulling out, like. So if you can breathe better through your nose, that's a positive Cottle Test. Some people you might not have it, but I definitely had that. And then that's related to nasal valve collapse or nasal valve insufficiency, however you want to call it. And that means your nasal valves are, when you're breathing in, there's not enough tightness in the cartilage here, so they collapse, and then that's causing airflow restriction through the nose. and can cut your sleep.
Yeah.
Can cause sleep apnea, can cause upper airway resistance syndrome with high RDI results on like WatchPAT or whatever.
Do they actually do surgery on it?
You can do various types of surgery. But what they do here is in the procedure I chose, Vivair. It's a customized Vivair procedure with some add-on stuff. They radio frequency 60 degrees to like really, it's like skin tightening but for your cartilage. So like when I do an Exilis or Exion procedure on someone, where you're doing radio frequency to tighten the skin at like 42 degrees roughly. It's a similar concept but higher temperature, around 60 degrees I think, where they're doing it on the cartilage on the inside, and then that tightens and shrinks the cartilage, so you get more, physical resistance to nasal valve collapse, and prevents them from collapsing inwards, so that the Cottle Test is no longer making a significant difference in your nasal breathing. That was the first thing, and then the next thing, they stick something up there, then crack your inferior turbinate so that it moved to the other side, so there's more airflow direction.
Painful?
And then they shove the radio frequency in there to shrink it as well. It's like sticking a needle into a hot dog and then sucking out the hot dog, and then that shrinks it. Kind of concept.
Is it painful?
Nah.
Oh.
Lot of salmon sperm is way more painful. Salmon sperm is like, because in this one they're doing a lot of I think it was xylocaine for the nasal surgery, and then that was like, I didn't feel anything really.
So tell us how to live forever. What do you do?
Fix your nose.
That is a big one, honestly.
Don't breathe through your nose.
Then it affects your sleep, and I think that's probably a huge factor.
It gets worse with age as well. So sleep, your apnea hypopnea index and your respiratory distress index, which are two values everyone should know. TBH. Never mind your kind of, I don't know what's popular these days.
Plasma exchange therapy.
Yeah, exchange. What the Christ?
So just get better sleep. Make sure you have good.
Well, no, you should.
Airway.
Instead of knowing your kind of, you should know your basic blood tests, right? Mm-hmm. But you also want to know your AHI and RDI, because this just screws so many people, destroys their lives.
What does that stand for?
Apnea hypopnea index.
Okay.
That's the sleep apnea, and RDI, respiratory distress index.
And how do you test those?
WatchPAT device, order online.
Yeah.
Don't need anything. It's like $150. Mm-hmm. There's another device now where it's like a different one where you can do it for 7 days in a row for testing these things. Okay. But yeah, WatchPAT has.
I think a lot of people. I was just listening to a podcast, The Sleep Doctor, and I think a lot of people struggle with that when you gain weight too, the tongue and everything. And if you're not getting good quality sleep, if you're waking up multiple times throughout the night because, you're definitely not going to be extending your longevity.
Well, it just causes everything else. Causes heart disease, causes diabetes, causes weight gain, causes anxiety, causes depression, causes lack of muscle gain, causes low testosterone, causes perimenopausal symptoms, causes everything.
Yeah, sleep is super important.
And it's something that can be fixed as well. That's the thing. Like a lot of like fixing Alzheimer's, it's very experimental, right?
Yeah.
Works sometimes, but it's not like we have a clear pathway. Whereas fixing nose and sleep apnea, we have very effective therapies now. Yeah. Like the surgeries are really good.
You just did. Never heard of what you just did. But that sounds amazing.
Most people don't know about it. Like most
people
doctors
just go to surgery. They want to do what is it called? The
rhinoplasty.
Yeah.
Yeah.
They just.
There's a big disconnect of like doctors with sleep disordered breathing. Yeah. Like a lot of kind of like with and reflux as well. Kind of it's also related because it can cause sleep disordered breathing. Yeah, there's just like a lot of doctors don't know how to treat reflux. And they don't know how to treat sleep apnea.
Okay.
Let's do the CPAP, right, which isn't fixing the anatomical cause.
Yeah.
Or BiPAP,
Not the real root cause. So what is your typical day of longevity? Do you care about your longevity or just
Oh no, yeah. I'm pretty, typical day. Well, I wake up.
Red light, sauna, supplements.
I do supplements, supplements.
Are you the typical biohacker?
No, I like sauna. Just kind of, think I'll go to the anatomy one soon. I think you been?
Mm-hmm.
Do you like
No,
it?
I haven't been
Oh,
actually, but I definitely know of it.
yeah, yeah. I think that's good.
Yeah.
Just the sauna in the place right now is a bit. It's not. It's not vibe. It's not
Yeah.
vibe. It's been.
I'm
Bit
gonna go to
disgusting.
anatomy.
Yeah, I'll wait for anatomy. Yeah, sauna I like, but
What's
yeah,
your
I li-
favorite biohacks? What's your diet? What do you eat? Do you eat meat?
yeah, I eat
Okay.
everything kind
Okay.
of. But it's all about moderation and what feels good for you as
Yeah.
well,
Yeah.
and what doesn't fuck your. Well, actually, so I'm on the Acid Watchers diet.
What's that?
Low, basically foods below pH 4
Oh,
you
interesting.
don't eat, and pH 5 you don't eat for 1 month, and then other
Oh,
triggers of lower esophageal sphincter irritants, you cut those out as well, or like at least reduce them.
are you feeling good on it?
Yeah, cured my reflux, cured
Wow.
my like everyone I've tried it on is cured their reflux
Oh,
when they've
interesting.
been on like PPIs for whatever,
Wow.
or like just
Wow. Have you done you've
stop
done your
eating acid.
DNA testing? Your genes? You've done all your genes?
Yeah, they're pretty good.
But do you also base your diet off of your genes, your gene variants?
I mean, I got one. Yeah, I'm heterozygous for MTHFR, but the evidence is really not clear on that. Um, but it doesn't really cost much to take. It's
Methylated.
basically the same price to take methyl B12 or
Yeah.
folate
Yeah.
these
Yeah.
days as it is not.
I just
So
have some like I might have
I
a
do
harder
take
time absorbing iron. There's like
right.
just certain
I don't know how well that. So I don't really like a lot of genetics GWAS stuff because they're not FDA approved
okay.
for pharmaco.
So you think it might
Uh,
not be that accurate?
nutrigenomics, pharmacogenomics, like a lot of these things aren't really clinically useful or provable. What's better is just to rely on the phenotype in a lot of this stuff. There are great exceptions though, like ApoE, BRCA1, rare diseases, BRCA2, PARC1, PSEN1, APP. There's loads of exceptions, right? These are all clinical grade diagnostics, but so much crap in this GWAS space. Yeah.
So, okay, so your typical day, you eat some oatmeal, you eat some porridge, some nutty pudding.
Yeah, yeah. Oats are a bit annoying. They're always like, I don't
Yeah, I
know.
don't do oats anymore. Well,
Yeah.
it's more of a glyphosate thing, I think, but I haven't done
Oh,
oats in a
really?
long time.
Even yeah, don't trust the
They're
organic.
highly highly with
It's like
pesticides.
fake organic.
Yeah,
kind of.
yeah.
Mm. Oh, but
Cross
there's
contamination
the new
too.
label though, isn't there? Like
The
glyphosate
safe free.
free label
Yeah,
plus
yeah.
organic.
Yeah.
Do you think that's any good? I don't know.
If I'm outside the U.S., I feel safe, but like here, I don't
Yeah,
feel safe.
yeah. Yeah, it's true. You don't
So
really even the glyphosate free label. It's kind of like, do I really trust this?
yeah, that's true too. I
Yeah,
don't know.
I don't
But
know.
organic's a whole nother thing too. It's like, do I trust organic? And then I was interviewing the glyphosate girl, and if it's organic from overseas, there's not really that many regulations. If it's organic here, there's more regulations. So it gets very complicated. Then you have organic pesticides. It goes on and on. So
Organic pesticides that like punch the
they're not
insects in
gonna
the
kill
face.
you and give you cancer, but they're just gonna give you a little
I
just
was in Spain recently, and they used honey.
for pesticide. Like, wow.
Yeah, all the insects just fly into the honey pot and then they die.
Why not just give an easy solution? I don't know.
No.
Let's just
Honey's
break-
expensive.
Anyways, okay, so typical protocol: eat whole foods, you get good sleep, nasal passage.
I mean, there's so much. There's like, yeah, it's literally like, this is why I made the school, because it's just,
Okay, I need to check.
even I
this
can't remember
out.
this stuff. It's like,
Okay,
I use it for myself. That's
Okay.
partly why I made it, because
Oh.
like, it's
I need
just,
to check this out.
med school isn't enough.
Yeah, for sure.
It's just like,
Yeah,
it's too much. It's too much out there.
obviously.
But
Okay,
yeah,
so
especially airway, like AHI, RDI. If you don't know those numbers,
that's a good one. Yeah, I know the sleep doctor. There's the machine. He has it on his website. So if anyone wants to check it out,
Oh, nice.
that you
Yeah.
can order the sleep apnea testing device on his website.
Yeah. What
Yeah.
I do is I just like use like any LLM and ask it to find the cheapest provider of
Oh.
WatchPAT,
Okay.
and it'll find like who's got the best offer on. I think you can get like $130 now rather than
Oh, wow.
200 in some places I checked. And then you use the Coupert extension on your browser, like Chrome, and then automatically it'll try 10, 20 coupons and find the cheapest one for you.
Okay.
Good tips.
So where can everyone go find you? And tell us about how to find all your different companies.
I think that most of them are listed on my website. Yeah, just my website.
Okay, you don't want to. You have your clinic in London, your skin clinic,
Yep.
and then your gene therapy is down south.
Gene therapy is loading.
Helix. Okay.
It's loading. Yeah.
Okay, and then
Helix,
your
I'm
longevity school.
advising a little bit, but yeah, I'm starting something separate from that.
Okay, and your website is
Just my name.
what?
.com.
Dr. Oliver Zolman.
OliverZolman.com.
Okay. So then people can go and do the longevity school, and not be scammed.
Yeah, I honestly, you'll probably, yeah, I think most people over the course of a few years, they'll save more money from avoiding scams and getting discounts and stuff than they would have had they
They
not
didn't
purchased
take
the
it.
school.
Okay.
Yeah, so
Definitely. And then you're also working on something with, what is it called? Mousespan?
oh yeah,
Yeah,
so many projects.
I know.
Yeah, this is like analogous to Aubrey's project where he's doing combination therapies, like up to 10 different therapies at the same time in mice to see which combination extends their health span and lifespan the most. So similar concept, but slightly different strategy.
Okay. And so you're raising money for that as well. And do you have a website or anything up for that yet?
Yeah.
Okay.
Yeah.
Okay. So I just
All
wanted-
linked from my main website
Okay.
to all the little projects from there.
Amazing. Well, thank you so much for coming on the podcast.
Yeah,
I'm sure
thanks
we
for
could
having me.
talk for another 8 hours or 48 hours. I don't know. But I really appreciate you taking the time out of your day and coming on the podcast. So thank you so much.
Great.